Hello students, this video explains the structure and functions of ribosomes. So ribosomes are small organelles of cell which are having a dense features and has a role in protein fabrication. These are made up of nucleoprotein having their origination in nucleolus. The main components of ribosomes are proteins and RNA. So ribosomes are present mainly on endoplasmic reticulum and also they are seen scattered in cytoplasm. These are significant organelles present in the plant and animal cell. So I have shown you the picture of ribosome with small unit and the larger unit. In between you can see the mRNA. George Palade, a cell biologist from Romania, discovered this ribosome with the aid of an electron microscope and stated that ribosomes are granules or dense elements, and he awarded Nobel Prize for that. In 1958, Richard V. Roberts gave the organelles its name ribosome, and in 1970s its molecular structure was discovered, and however in 2000 a more perfect structure has been discovered with the help of microscope of higher resolution. Where these ribosomes are present. The occurrence of ribosomes, you can find them free ribosomes as they travel independently in cytoplasm, and the ribosomes are also seen membrane bound. They are present on endoplasmic reticulum. Here you can see the enlarged view of a cell, and you can see the ribosomes which are present on endoplasmic reticulum and also free floating ribosome. They are mainly composed of rRNA and protein. So what are the structural features of ribosome? The diameter of ribosome is 20 nanometer. Their composition can be divided into two parts. 2/3 part of rRNA or ribosomal RNA and 1/3 part ribosomal protein or ribonucleoprotein. The structure of ribosome and its subunits are almost similar for both prokaryotes and eukaryotes. The ribosomes in case of prokaryotes are 70S, whereas where each one comprises 30S small and 50S large subunits, and 50S further comprises 5S subunit of RNA comprising almost 120 nucleotides along with the subunits of 23S RNA and composed of almost 2900 nucleotides, and 34 protein subunits, and 30S is evaluated. If the 30S is evaluated, subunit of RNA it comprises about 1540 nucleotide. This notice with are bounded through 21 units of protein. Ribosomes in case of eukaryotes are 80S, where each one comprises 40S small and 60S large subunits. Okay, it is present in the larger subunits. 60S comprises 5S subunit of RNA, that is almost 120 nucleotides along with the units of 28S RNA. They almost have 4700 nucleotides. Subunit of 5S close to 160 nucleotides and approximately 49 units of protein, and 40 as 80S RNA approximately 1900 nucleotide, and approximately it is 33 units of protein. So here I have given the difference between prokaryotic ribosome and eukaryotic ribosome. Prokaryotic ribosome it is 70S ribosome. Eukaryotic ribosome it is 80S ribosome. In case of prokaryotes, the largest subunit is 50S and smaller subunit is 30S subunit. In case of eukaryotes, it is 80S and 40S. There you can see 5S RNA, 23S RNA and 16S RNA in case of prokaryotic ribosome, and in case of eukaryotic it is 5 RNA and 5.8S, 28S and 18S RNA. So these RNAs, what I am that is located here, they have the role in the attachment of tRNA and mRNA during the process of protein synthesis. These ribosomes, they are not always associated. They undergo a cycle of association and dissociation. Okay, so here you can see the smaller subunit and the larger subunit, the separated subunits, and these are the second picture. It shows the unit, the whole. It is a union of both the smaller and larger subunit. What are the functions of ribosome? The sole function, it is protein synthesis. It is a prominent function of ribosomes. The RNA contents of ribosomes are transformed into protein molecules. The cell, which requires a numerous protein for its proper functioning, all the cellular proteins, they are synthesized in the ribosome. It is the organelle that coordinate together to carry out all cellular activities. So this is the picture shows the translation process. Here you can see the smaller subunit, larger subunit, and with the mRNA. So mRNA, it is the codons which are present in the mRNA, they are decoded in the ribosomes. So according to the codon, the tRNA molecules brings the new amino acid residue, and the new amino acid residue, it forms a peptide bond with the already present amino acid at the site of the peptidyl transfer. That is we also called as P site. Okay, so at a time the ribosome can accommodate 3 tRNA molecules. That is one that is accepted and 1 tRNA present at the site of that is peptide bond synthesis, and the other one which exits from the site of the ribosome. Okay, so growing polypeptide chain also you can notice. As I told you, this ribosome, it has a 3 important sites, that is EPA. What is E site? E site is the exit site. Okay, P site is the peptidyl tRNA binding site. We call it as P site, and the A site, it is amino acyl tRNA binding site, where the new incoming tRNA brings the amino acid to the site of protein synthesis. And here you can see the site where mRNA binds, okay, and the larger subunit and the smaller subunit. So this is the overall picture how this protein synthesis takes place. So from the DNA, the information, it is transcripted in the form of RNA. Okay, so mainly it is mRNA. In case of eukaryotes, mRNA undergoes some post-translational modification, and this mRNA is transported from nucleus to the cytoplasm. In cytoplasm, mRNA undergoes translation when the single mRNA can be translated by many ribosomes. So during translation, where all the codons are read by these ribosomes, and they will result in the formation of a polypeptide chain. This polypeptide chain, as soon as it is synthesized, it is called a just born or nascent polypeptide chain. Again, this polypeptide chain undergoes some modification. So that those modifications, we call them as post-translational modification. Again, they occur in the lumen of endoplasmic reticulum. So after post-translational modification, what you get is a mature protein. So it has its function. So the overall the overall picture, it depicts the role of ribosomes inside the cell, mainly the protein synthesis. So in summary, a ribosome is a complex of RNA and protein and is therefore known as a ribonuclear protein. It is composed of 2 subunits, smaller and larger, and ribosomes are the organelles that help in protein synthesis, and protein is required for all cellular activities such as damage, repair, and other chemical or metabolic reactions. Okay, so this is the overall summary of ribosomes. This is the structure and functions of ribosomes. Okay, thank you for watching.